study are the identification of some ecologically important amphibians. For example, the Philippine endemic Platymantis guentheri. In terms of reptiles, a very interesting one example is the Lycodon dumerilii, which exhibits a widespread distribution and has been documented throughout the Mindanao Pai or the Pleistocene Aggregate Island Complex. Ms. Elaine discussed also about her interesting findings in birds. For example, the Bucerus hydrothorax semigaleatus, also known as the Philippine hornbill, a large species of hornbill endemic to the Philippines. In terms of flora, she found this new island wreck species, for example, the Victoria calcarea, a fern or herb species with native range in the Philippines. Overall, Ms. Elaine emphasized that SINP is indeed worthy of its nomination as a UNESCO Natural Heritage Site because of its natural and cultural significance. The following month, Ms. Renty Ovenia successfully presented a very good study on some threatened woody plant species of forest of the limestone in the Philippine Islands. The study determined the threatened woody plant species in the forest of the limestone in the Philippines and discussed the threats of the forest of the limestone in the country. And of course, Samar Island is one of her study sites in addition to Cebu and Batangas sites. Ms. Ren presented the list of critically endangered species based on the Philippine DO Red List and the IUCN Red List. She also made an emphasis on economic uses of some of the identified threatened woody plant species in her study sites. For example, the Sharea nigrosensis, a Philippine endemic pterocarp species, and our very own Ana philippinensis, with no natural populations in Batangas and in Mindoro. Third one is the Aquilaria cumingiana, a source of agar root, a highly prized resin with many economic values. And for example, is the Opea philippinensis, which is also a Philippine endemic dipterocarp species. Part of the study is the identification of threats and conservation concerns in the study site. For example, the limestone quarrying, deforestation, illegal logging, among others. Ms. Ren concluded the presentation by highlighting the potential contribution of her study to sustainable development goals. In May 2022, UPLB Assistant Professor Desamarie Antoinette Fernandez gave us new insights into biodiversity of forest over limestone in Caliguan Island, Iwan, Eastern Samar, Philippines. She also did biodiversity assessment in the study site for both flora and fauna. Interestingly, Assistant Professor Fernandez recorded 41 plant species belonging to 18 families, of which 5 species are Philippine endemics, 9 threatened species based on the IUCN Red List, and 5 threatened species based on the DAO 2017-11 Red List. And these are some of the photographs of the plant species he identified. Walashodendron salabicum, which is locally known as panuyo, Aquilaria cumingiana, which is one of the agar-wood-producing Aquilaria species found in the Philippines, and Hansea wenzeliana, also one of the identified endangered species based on the IUCN Red List. For fauna component, 21 vertebrate species were recorded, of which 6 are documented throughout the Mindanao Pike or the Pleistocene Aggregate Island Complex. And here are some of the photographs of the vertebrate species he recorded from the study site. Just last month, we learned a lot from UPLB Assistant Professor Marjorie de Los Angeles, who discussed about the ecology of understory plants of forest over limestone in Samar Island, Philippines. She documented plant understory species in forest over limestones and described a pattern of plant distribution with reference to ecological parameters using multivariate tools in Paranas, DAC, and E1 Samar. Assistant Professor Marjorie recorded 26 understory floral species divided into 21 flowering species and 6 fern species. Part of her study is to determine the richness and diversity indices of Paranas, DAC, and E1. In addition, she found that the understory plant species from Paranas and DAC sites are highly affected by soil iron concentration and electrical conductivity, whereas plants from B1 are highly associated with soil pH and other macro and micronutrients.
Samar Island is the third largest island in the Philippines and has one of the most extensive forest over limestone ecosystems in the country. This ecosystem is home to a wide array of uniquely adapted flora and fauna. Samar Island is known to be one of the centers of botanical diversity in the Philippines and has been nominated as a UNESCO World Natural Heritage Site for its exceptional natural beauty and precious biodiversity. For these reasons, CONserve-KAIGANGAN, a biodiversity research program, was established. Kaigangan is a Samar local word that means forest over limestone. Our research program's mission is to assess and conserve the biodiversity in Samar Island's forest over limestone towards its sustainable management. One of our program's projects is the assessment of biodiversity and community structure in forest over limestone ecosystems. This component of our research aims to determine plant diversity and vegetation structure in the various habitat types in forests over limestone, to determine animal diversity associated with forest over limestone, to identify functional classification of vegetation with its associated animal diversity, and to analyze the environmental factors influencing the prevailing structure of the biota. We conducted a comprehensive biodiversity assessment of forests over limestone in the municipalities of Paranas, Taft, and Giwan with remarkable findings. A combined total of 71 plant species were recorded from these municipalities, with at least 3 tree species classified as vulnerable based on the DENR updated national list of threatened Philippine plants. A total of 52 terrestrial vertebrate animal species were also recorded, including 5 amphibian species, 15 reptile species, 20 bird species, and 12 mammal species, with conservation statuses ranging from threatened to endangered and critically endangered, based on the DENR updated national list of threatened Philippine fauna. This knowledge will empower our stakeholders in these areas to make appropriate research-based conservation efforts for the protection of these ecosystems that harbor an assemblage of rare, endemic, and economically important flora and fauna. Through this program, people will be equipped with knowledge on forest over limestone biodiversity, including the ecosystem services that it can offer to the community. We at CONserve-KAIGANGAN continue to further raise awareness and capacitate national and local government agencies, especially local communities living in and around Samar's Kaigangan.
Welcome to our Kaigangan Biodiversity webinars. I am Nova Hilvano, your host for today. This webinar aims to raise awareness on the conservation and sustainable use of Kaigangan biodiversity through a series of online talks from our very own experts. This webinar is made possible by CONserve-KAIGANGAN, a research program implemented by the University of the Philippines, Los Baños, Samar State University, and Eastern Samar State University. This program is also funded by the UST PCARD. Please feel free to greet everyone and introduce yourselves through our chat box. Please include your organization and location if possible. We have participants here coming from Cebu, and we also have participants from Eastern Samar. Good morning po. So before we begin, we would like to remind you of some of our guidelines. First, please keep your microphones muted. Second, you may send your questions using Zoom or chat. We will accommodate your question. Please certificate. Please fill out our evaluation form. The Google form link will be posted in Zoom and Facebook chat box at the end of our program. These certificates are sent the email indicated in the evaluation. So, but for this webinar. Okay po, I'm back. We really have a good internet connection here. So our speaker this morning is a assistant professor at the Samar State University. She had been in the service for 20 years and she is a licensed teacher. Our speaker took her elementary education at the Osmeña Elementary School, her high school education at the Leyte National High School. She took her college and master's degree at the Leyte Normal University. Her bachelor's degree is secondary education, and her major in biology, and her master's degree is Master of Biology. She finished her Doctor of Philosophy, major in Science Education at the University of Perpetual Health System, Delta. Our speaker is designated as the Natural Science Chairperson, Chemistry Lab in Charge, and Extension Coordinator of the College of Arts and Sciences at the Samar State University. At the same time, she is the Laboratory Manager at the Analytical and Biochemical Laboratory of the Research and Innovation Center of the same university. Our speaker is also an active member of several professional organizations. She is a member of the Philippine Organization of Science and Technology Educators, and also a member of the Philippine Association of Research Managers Incorporated, State Universities and Colleges Teachers Educators Association, Biology Teachers Association of the Philippines Incorporated, Federation of Institutions for Marine and Freshwater Sciences, and Samar State University Personnel Association. In addition to that, our speaker is also a researcher and presenters in scientific conferences. For example, she presented during the first International Conference on Poverty Alleviation and Sustainable Development, and during the second International STEM Research Congress. She also leads institutionally funded research on physical, chemical, and microbial analysis of waters in Samar mussel farms. She also conducted the following researches: antibacterial activity of methanolic leaf extract of Cristela species against human pathogens, development and evaluation of wound healing ointment with fishbone and firm extract, development and evaluation, spectrometric analysis of anthocyanin from purple sweet potato, antioxidant activity of flavonoids isolates from leaves of Cristela dentata, and WatchHub Region 8 warning and technology to combat hazards of harmful algal blooms in Region 8. In the Project 2, this involves the understanding the social dimension of red tide phenomenon in summer. She is also involved in the development of plant-based mudcrab feeds with probiotics. She also published scientific paper on physical, chemical, and microbial analysis of seawater in summer mussel farms in an ISI Scopus referred journal. She also published the following utility models: coconut wine with wild honey, process of making antibacterial soap bar with Cristela species extract, antibacterial soap bar with Cristela species extract, process of making acid-base indicator from Moses in a basket extract, organic fertilizer derived from green mussel shells and Philippine orange, process of making organic fertilizer derived from green mussel shells, Latundan banana peels, and Philippine orange, rodenticide fillet from makabuhay vines, extract, and tugi root crop grate, process of making rodenticide fillet from makabuhay vines, extract, and tugi root crop grate. In addition, she also holds several copyright registration and deposit at the National Library of the Philippines. This includes T-learning worksheets in botany with laboratory activities, semantic diagram in history of botany, registration, physics, mechanics, and heat learning, and Killing the Green Mussels 2.0, which is a video documentary. Ladies and gentlemen, let us all welcome our speaker this morning, Professor Edelyn Echapare. Ma'am.
Hello po. Hello sa lahat. Thank you, Ms. Nova, for that napakagaling ng pagkaka-introduce. Thank you very much. So hopefully, I can share something later in my video presentation. And I would like to thank Dr. June Buot for inviting me to be part of this Kaigangan project, which is also my interest. Thank you very much.
Hello everyone, I am Edelyn Echapare. I will going to share with you about a certain plant that can be found in Kaigangan in Samar. This plant is formerly used or utilized by the people merely for ornamental, but through scientific exploration, this plant has become economically important. To start with, shown in the picture is a plant taken from Kaigangan in Samar, and were submitted to National Museum Botany section for its proper identification and authenticity. And it has been found out to be Cristela dentata, which is the rhodophyte that belongs to the family Telepteridaceae. It is a soft fern with long green frond. Another fern plant that can be found in Kaigangan in Samar that belongs to the same family with that of Cristela dentata is Macroptelepteris. This particular fern plant is a perennial fern that grows in disturbed areas, including roadsides and forest edges, and behaves as a weed. It spreads via spores in wind and water, but can also be spread via stones and rhizomes in garden waste. The Lepteridaceae are abundant species in summer, but they underestimated because of their livelihood potential and economic needs. So we get interested with this plant. From this, we started scientific exploration of some fern plants. We were able to analyze the phytochemical content of the plant and also examine its antibacterial properties. We collected samples from Kaigangan and brought these samples to SSU laboratory for antibacterial evaluation and phytochemical screening. For the phytochemical screening and extraction, a particular solvent has been utilized in the extraction process. Specific solvent, for example, if the test is for external CONserve-KAIGANGAN, alcohol is used as solvent in the extraction. For the antibacterial properties evaluation of the plant sample, we use the disc diffusion method in which the zone of inhibition is the reference, how susceptible a particular extract to bacterial growth. And for the results of the conducted experimentation, the plant extract contains saponin, tannin, organic acid, and alkaloids. It was also proven that the plant extract has an ability to prevent bacterial growth, either gram-positive or gram-negative bacteria. Statistically, the antibacterial activity of the plant extract has no significant difference with that of either gram-positive or gram-negative, and with that of the positive control. So this result entails that the plant extract has the potential to prevent bacterial growth. From the result of the study we conducted, we come up with a product. So here comes the fern soap. Fern soap is the germicidal with the extract added to the fern extract, and the product has a moisturizing effect. The physical chemical of this blend was conducted with the formulated food. For instance, the weight loss on curing. The samples were allowed to cure at room temperature control during the scientific phase of the sample. Were taken after every week interval. So the weight loss was determined by subtracting the initial weight to get out the weight of the sample every 7 days. Another physicochemical parameters that has been evaluated were the moisture content, foam height, and foam retention. Moisture content was measured using the thermogravimetric method. With this method, the moisture is determined from the weight loss of the sample, dried by heating, in which a 5 gram of each sample was run in the moisture analyzer balance was set at 160 degrees Celsius, and the rate of moisture changing 0.05%. For the foam height, The 1 gram of the soap sample was dissolved completely in 100 ml of water. Then take the 10 ml of the solution into the separate boiling tube. Close the tube with a rubber cap and shake the tube vigorously for 3 minutes. Let the solution set for 10 minutes and measure the foam height. So we recorded the observation for 3 consecutive experiments, and then it was able was computed. And then for the foam retention, time refers to the time for which the foam produced by the soap retained. So it has been the above procedure has been repeated, and the foam interval was measured for about 5 to 10 minutes. Other attributes of the soap were also evaluated using the sensorial evaluation. We made a questionnaire. We utilized the sensory 9 hedonic scale evaluation for gathering participants' preference as to fragrance, foaming, rinsing, moisture, irritancy, and overall acceptability of the soap.
Thank you very much, Professor Echapare, for sharing with us the scientific exploration, utilization, and commercialization of ferns in Samar's Kaigangan. So if you have any questions to Professor Echapare, please leave them in our Zoom chat box or in the comment section of our Facebook Live. Receive an e-certificate for this event. Please do not forget to fill out our evaluation form, which will be posted later. So we will now proceed to answer some questions from our audience. To help us with our Q&A session, I would like to call on Ma'am Ren Ovenia, the University Research Associate of the CONserve-KAIGANGAN project. Ma'am Ren.
Good morning po. Good morning, everyone. I am Ren Ovenia, current University Research Associate of the CONserve-KAIGANGAN program. Welcome po to our webinar and thank you for attending din po. So if you have any question kay Ma'am Edelyn, you can type it in the Zoom chat box, or we are streaming via FB Live, so you can comment your questions to Ma'am Edelyn. So without further ado, may I call po Ma'am Edelyn Echapare to answer some questions already sent by our participants. Ma'am Ed, good
Opo,
morning.
sige po. Good morning po.
po. So the first question po is, aside from the mentioned commercialization, are you or the team planning to develop it as a small-scale business or a large-scale business?
Actually, ano po siya, kumikitang kabuhayan. So, I1 is, the group or the team established a company which has been issued. One of the spin-off company of Samar State University, which is all natural, na yung pangalan ng company. And this particular company is owned by kami pa rin po. Tapos yung we exclusively distribute or kami po yung nagbibenta ng fern soap po na ginagawa. Kami din po yung gumagawa.
Ah, so ma'am, ongoing po yung pag-produce po ng products po ninyo na soap?
Opo, opo. May
Until
mga
now po?
may mga nag-o-order na din po. Tapos the TBI Lambat here in SSU, which is a business incubator, sila din po tumutulong na madala sa market yung product po. So kung saan-saan na po nakarating yung fern soap namin all over the Philippines. Baka dito lang sa Samar hindi. Pero nakarating na po sa at least in Luzon and then in Mindanao. Opo.
Ma'am, kailan po nag-start po yung pag-produce po ninyo ng soap and then kailan po siya na-release sa public?
Bali po yung massive talaga na production, pinakamarami nung kasagsagan ng COVID. Kasi
Okay.
one of the project po ng which is part of the SSU Bantay COVID na projects is a production of the germicidal fern soap, which is funded by CHED. So yun po, we produced 6,000 bars and we were able to distribute this 6,000 bars for free para sa mga employees of SSU and students po.
Okay po, ma'am. Yung next question po is, tinanong po is the product can be produced household po or does it require parang high-end laboratory equipment to make the soap? And if, opo, yes po, if the locals are in
Pwede po siyang gawin sa bahay, pwedeng homemade po.
okay po.
Opo, yung kailangan lang ay yung mga molders at saka yung stick blender lang po yun. Stick blender kasi kailangan po yun for the homogeneity of the mixture. Opo. Kasi hindi siya kaya ng basta stirring yung mga sandok ganon, hindi siya kaya. Dapat talaga mechanical stick blender ganon. Pero this can be made at home.
Okay po, ma'am.
Opo.
Ma'am, ang next question po is regarding naman po sa monetary. So, usually po, per batch po ang paggawa, no, ma'am? So, for
Opo.
example, for isang batch, parang ilang mga pieces po ang napoproduce? And then, how much will it cost? And then, what would be the profit if ever po yung mga household natin is planong magbenta po ng product na ito?
Ay, opo. Ang tawag dito? May calculation tayo. Pero yung cost estimate po namin, bawat 90 bars, it cost, yung production tsaka kasama na yung mga materials, it cost 14 pesos. Tapos pwede siyang maibenta ng 25 pesos po. Mga 90 grams po yun.
Wow, wow. Ang mura po pala ng
Opo. So you can start very small amount. Opo. Tsaka din po kami, nag-conduct din po kami ng mga extension activities na tinuturo po namin yung paggawa po namin ng sabon, bali as part of technology transfer na din po dun sa mga people's organization na may gusto na ma-train sila para magkaroon sila ng livelihood or skill na ganun as part of their livelihood activity po.
Okay po. So, ma'am, yung yung product po ninyo is patented na po ba siya? Or parang are you planning to
Opo.
apply po? Okay po.
Opo, nakaano na po siya. Bago pa po kami mag-produce, bale, na-register na po namin siya for registration sa ITOFIL.
Okay po, ma'am. So, ang next question po is regarding naman po with the plant collection samples that you use in the products. Are there any pre-processing procedure po before extracting the elements of extracting the elements that you needed from the plant species?
Ay, opo. Kasi kailangan po siyang i-clean, washed with water before, actually sun drying pwede rin siya. pwedeng air drying, pwede rin sa oven po. Trinay namin yung tatlo. Parang ganun din po naman yung effect. So it's either sun drying, air drying, or sa oven po din na dry. So yun
Ah,
po. Before finally it will be extracted.
okay po. So ma'am, bali po, hindi po necessary na fresh yung samples na gagamitin. Bali, ide-dry pa po siya.
Yung purpose po ng drying is ano po, para matagal po. Pero trinay din namin yung fresh. Parang wala naman po siyang significant parang significant difference. Parang nandun pa rin po yung bioactive material niya na preserve pa din po. It's either gagamitin siyang fresh or dried na.
Oh, okay po,
So yung
ma'am.
purpose po ng drying para ma-keep po namin siya ng matagal.
Okay.
Opo.
So, next question po is, aside from soap, have you used the plant species for other products po?
Sa ngayon wala pa po, pero may mga ibang researchers din po dito sa SSU, which is gumagamit din po sila ng fern, pero hindi po yung particular na fern na ginamit namin dun sa sabon. Bali yung the product that they produce is parang oil for parang pang spasm.
Oh,
Ini-extract nila yung oil from a particular fern. Fern din siya, pero hindi siya yung Cristela dentata. Yun po. Pero yung sa amin po, sa ngayon, yung soap pa rin po.
okay po, ma'am. So, ma'am, pero ngayon po, yung aside po from the mentioned species, are there any ongoing studies or current scientific exploration on the other plant species found in Samar's Kaigangan?
Bali yung sa grupo namin, focus po muna kami dun sa Cristela dentata kasi
Okay po,
yung
ma'am.
ina-after po namin, parang i-extract namin yung particular talaga ang tawag dito, yung bioactive material na nandun sa Cristela dentata, that what makes it very effective in what's this, in preventing bacterial growth. So alin ba talaga doon sa mga na-identify namin? Kasi yung procedure na nagawa namin is just identification. Wala pa po yung isolation at saka
Okay
quantification.
po.
Opo, wala pa po kami doon. So as of now, doon pa rin po kami sa quantification and isolation if that could be possible.
Okay po. So, for the last question, na po, sorry for the cutting off na rin po ng time since we have a limited time po
Mm,
regarding with the
okay po.
presentation
Pero
for
yung
today.
yung mga gustong matuto, we are willing to naman po, extend our service para turuan po sila. I-contact na lang po kami dito sa SSU.
Ay,
The
okay.
team is willing to share with you what we know about soap making.
Opo. Ma'am, may nagpahabol pa po ng question.
Opa.
Last na po, ma'am. What are the advantages and disadvantages of using local plant species in a product?
Dun sa particularly dun sa product na ano ba yung disadvantage? Pero yung advantage kasi, di ba, locally available siya. Tapos mas low cost yung production kasi nga nandiyan lang siya sa paligid.
Yes po.
So parang we do not have to spend much for us to have the product, lalo na dun sa production. Opo. Yung disadvantage. Hindi
Iniisip
ko
ko
ba?
din kapagka po halimbawang endemic 'no, tapos
Oo nga.
siguro po dapat, yun lang po yung siguraduhin po muna kung yung conservation status pa niya,
Ay,
determine
opo.
yun po ma'am 'no. Tsaka
Opo,
po
yun
for
po. Kasi baka
example
madami na yung gawing
opo,
sabon,
ma'am,
makalbo na.
makalbo na po, yes po
Ay,
ma'am.
opo, opo. Kasi
Yes.
actually may ginagawang kung pwedeng i-culture siya, gano'n. Parang may
Opo
nakikipag-collab
ma'am,
sa amin
yes.
na ibang university. na parang i-culture po yung particular na fern para hindi naman siya hindi masyadong makalbo.
Oh,
Opo,
okay,
opo.
ma'am. Ang galing po. Tissue culture po ng ferns. Isa pong way
Opo.
ng yes po. Okay po, ma'am. Ma'am, I think yun na po yung lahat ng question. Thank you po for entertaining the
Yeah.
inquiries of our participants for today. And thank you, ma'am, for accepting our invitation to be our guest speaker for this webinar, ma'am. And then
Oh, thank you po
yes
sa
po, ma'am.
opportunity.
Opo. Ma'am, yun na po, na-mention niyo po last time na if they interested to have collaboration with your institution, would it be okay, or if they have any other questions about the topic you've presented, would it be okay po to contact you, or
Opo,
yes po, ma'am.
sige
Ayun po.
po.
May I ask na lang po na if okay lang po na i-post po natin sa chat box ang inyong email, if ever
Sige
po.
po, okay lang
Yes
po.
po, okay po.
Sige
Thank you very
po.
much, ma'am.
Sige
Thank you
po.
po.
Welcome.
Opo. I will send na po yung ask na po Ma'am Nova to take over and proceed po with the program. Thank you, ma'am.
Okay po.
Thank you very much, Ma'am Echapare. As you mentioned earlier, can you share to our participants your contact details, especially those who are interested to contact you? It's already there. Thank you very much po, ma'am. So we would like to extend our deepest appreciation to ma'am.
Will issue po the a certificate of appreciation to Ma'am Edelyn Echapare. For a while lang po, I'll share in the certificate. So, University of the Philippines España Certificate of Appreciation is awarded to Edelyn O. Echapare for serving as a research speaker during the webinar paper presentation entitled Scientific Exploration, Utilization, and Commercialization of Ferns in Samar's Kaigangan of the 2020 Kaigangan Biodiversity Webinar Series held on 28th of July 2022 via Zoom. Designed by Dr. Inocencio E. Buot Jr., Professor and Project Leader of CONserve-KAIGANGAN. So, thank you again po, Ma'am Eds, for accepting our invitation. So, back to you po, Ma'am Nova.
Okay po, thank you again. Ma'am Echapare, the link for the evaluation form is now shared in the comment section chat box, and please fill out the form to receive your e-certificate. Please be reminded that the evaluation form will be closed 24 hours after its posting. Ma'am Ren, please, for your closing remarks. Thank you po.
Before we end po yung ating program, may request everyone po to open their camera for group photo. Hello po. And hintay pa po tayo ng ibang participants na mag-open ng kanilang camera. Okay po. So, wala na po ba nga habol? Okay, slide 1 po. For a while po. Ayan. Okay po. Thank you very much po sa mga nag-open ng camera. And yun po, start na po ako. Slide 1. 1, 2, 3, smile. Isa pa po. 1, 2, 3, smile. Next, next part po ng participants. Okay. 1, 2, 3, smile. 1, 2, 3, smile. Thank you po, thank you po. Okay na po. So thank you everyone po for attending the webinar series for today. The link for the evaluation form is now shared in the comment section or chat box. So please fill out the form to receive your e-certificate. So please be also reminded that the evaluation form will be closed 24 hours after its posting. So kindly sasagot na lang po agad. Also, those who attended the Kaigangan Biodiversity webinar series from March 22 to December 2022 will receive another certificate. So don't miss out our future webinar. So, ayun. And allow me to share another. So, next, our next speaker for the Kaigangan Biodiversity webinar series for the month of August 2022 is Assistant Professor in University of the Philippines, Los Baños, Professor Marjorie de Los Angeles. She will be presenting the noteworthy floral species in forest over limestone of Samar Island. So this will be also held online via Zoom and Facebook Live. So we will post the registration form and other details of the webinar on our Facebook page. So that would be all. I will return now the stage to Professor Silvano. Ma'am.
Okay, thank you very much, our participants. Thank you also, Sir Buot, for gracing our today's webinar. So on behalf of the program team, we would like to thank all the participants who joined us in our event for 2022. Hope you have learned from our speaker and would be able to apply it. If you want to know more about the CONserve-KAIGANGAN program, you may visit our Facebook and YouTube page, or you can send us an email. The links are posted in our comment section and chat box. Please do not forget to fill out the evaluation form to receive your e-certificate for today's event. Please be reminded that the evaluation form will be closed 24 hours after its posting. Thank you, everyone, and enjoy the rest of the day. Thank you, po.
Pahabol lang po, you may also stay for a while after the closing remark video of the closing video presentation if you're interested to chat with us or to other participants. So, see you po.
Samar Island is the third largest island of the Philippines and has one of the most extensive forest over limestone ecosystems in the country. This ecosystem is home to a wide array of uniquely adapted flora and fauna. Samar Island is known to be one of the centers of botanical diversity in the Philippines and has been nominated as a UNESCO World Natural Heritage Site for its exceptional natural beauty and precious biodiversity. For these reasons, CONserve-KAIGANGAN, a biodiversity research program, was established. Kaigangan is a Samar local word that means forest over limestone. Our research program's mission is to assess and conserve the biodiversity in Samar Island's forest over limestone towards its sustainable management. One of our program's projects is the assessment of biodiversity and community structure and forest over limestone ecosystems. This component of our research aims to determine plant diversity and vegetation structure in various habitat types in forests over limestone, to determine animal diversity associated with forest over limestone, to identify functional classification of vegetation with its associated animal diversity, and to analyze the environmental factors influencing the prevailing structure of the biota. We conducted a comprehensive biodiversity assessment of forests over limestone in the municipalities of Paranas, Taf, and Giwan with remarkable findings. A combined total of 71 plant species were recorded from these municipalities, with at least 3 tree species classified as vulnerable based on the DENR updated national list of threatened Philippine plants. A total of 52 terrestrial vertebrate animal species were also recorded, including 5 amphibian species, 15 reptile species, 20 bird species, and 12 mammal species, with conservation statuses ranging from threatened to endangered and critically endangered, based on the DENR updated national list of threatened Philippine fauna. This knowledge will empower our stakeholders in these areas to make appropriate research-based conservation efforts for the protection of these ecosystems that harbor an assemblage of rare, endemic, and economically important flora and fauna. Through this program, people will be equipped with knowledge on forests over limestone biodiversity, including the ecosystem services that it can offer to the community. We at CONserve-KAIGANGAN continue to further raise awareness and capacitate national and local government agencies, especially local communities living in and around Samar's Kaigangan.